with specific doxorubicin and peptide, we could actually see that the tumor mass reduced through the 28 days compared to the non-application of magnetic hyperthermia. So therefore, drug targeting wouldn't work without an activation induced. And this is also linked up to the aspect that magnetic hyperthermia has also a physical effect that was demonstrated by other partners, which actually shown that the hyperthermia has a softening effect on the tissue, which actually brings to a so-called heating effect to destructive tumor matrix. So it actually releases the T-junction connectivity between cells, and through that aspect, you can actually have a better penetration, a better delivery of the drugs through time after application. Further into that, they investigated the difference between intratumoral injection and intravenous injection, and that was quite striking, the difference, since it prevents, intratumoral injection prevents all the other problems that we have seen by the intravenous injection, like accumulation, hemolytic response, procoagulation, and complement, as we just heard in the extensive in the previous. So this one show that deconstructing the tumoral matrix actually has an opportunity where the tumor growth can be diminished by the application of drug and nanoparticle, and that was effectively proven. We're still at the base of our NoCanTher. So that's where NoCanTher come in. We capitalize of all this knowledge, and we bring the particle, the NoCan, the drug, and the hyperthermia, and we mix them all into the opportunity to create a scale-up under the guidance of a pharmaceutical company, which is Biopraxis, which is not in this room because they presented in the room parallel to that at 11 o'clock, a talk about how they do clinical trial translation in their already ongoing products. So having all this expertise from the developer to the actual application, to the actual pilot opportunity, create a unique condition and environment where NoCanTher was funded for 5 years with the ultimate goal to go into clinical. Further into that, the combination with Val-d'Hebron and the expertise of CNIO as a Cancer National Institute brought the aspect that we can mediate with the regulators, and therefore we can actually expedite our investigation towards the submission of a dossier for clinical trial investigation. So the particles are iron oxide core, they are coated, and they have NoCan peptide on the surface. We're trying to keep them as simple as possible because we are capitalizing what the previous experience developed by one other partner, which is Resonance Circuit Limited, which they actually have worked with endomagnetics, which have the only iron oxide particle that has been FDA and EMA approved for injection. So through that, we have experience on the preparation of the particle. So the formulation has been already set from the perspective, and the work is actually developing into two fronts under NoCanTher. The first one is the good laboratory practice, where we have setting up a laboratory where we can do the full preparation in a sterile environment without having to comply in full with GMP. So it's good lab practice where we can do characterization and pre-assessment as preclinical study. So this has reduced the cost before you can go into the full batch production. And the creation of tools that can measure the specific absorption rate that the particle have to deliver for hyperthermia, for the opportunity to cleave the linkers, but also deliver the given temperature and reaching the combined temperature of 43 degrees for cancer killing. Further into that, the clinical investigation is going to be work through 3 steps. The preclinical studies are carried out within the consortium, but also in conjunction with the European Nanomedicine Characterization Laboratory. So we are at the fortuitous time where we can submit the case to the UNCL, and through that we can actually avail of this, and that will help us on the preclinical. The magnetic field generator for magnetic hyperthermia capitalize of 28 years of experience of one of the partner, which is RCL, which previously also have competencies through endomagnetics, so they are the best company to assist on the development on magnetic field generation. And the in silico magnetic hyperthermia studies are carried out as a means of computational modeling and also evaluation of the magnetic properties. The design of the clinical studies, well, we cannot be in a better place with Vall d'Hebron being the first university hospital that has launched Abraxane combined with gencitabine as a clinical trial in Spain. So therefore, they have already all the ethical and the pipeline for the development of the clinical study as a design. So where are we at in the roadmap? The project is only 1 year old. We have already made massive improvement towards the preclinical study, and we are developing with Biopraxis the pilot line. We were in Bilbao last week, and that's where we discussed about pilot line production of the particle through one of their sterile line. The second aspect is the regulatory aspect for the production have been already identified and they are in compliance. And the discussion with the Spanish regulatory authorities is already taking place since day one of the project. So we're not letting things slipping behind. And the clinical study should start by 2020. We have a set deadline. So we work with deadline as industrially relevant. Biopraxis will give a talk in the next section. Professor Schwartz and Ibane Basolo are here around if you want to have further discussion through that. The partner and the team is the most spread and competent. And we also are very active into disseminating. And we look forward to have all your students, your postgrad at the European Society for Nanomedicine in Barcelona in September, where there's going to be a NoCanTher session. And therefore, we invite everybody to attend the event since it's funded under the CLINAM umbrella. Thank you very much for your attention. I'd like to thank NoCanTher as a project, the chairman, and you for being here in the session. I'd like to take any question if there's any.
Thank you very much for this very elegant presentation. We do have time for questions. Yes, please.
Adriele.
Yeah.
Many thanks for the very interesting presentation. Could you please explain, do you know what particle size has the threshold of magnetic capability, of magnetic properties of your particles?
It's a very good question. Yes, we did, because it was matter of the study with Multifan. The particle size range in the core is between 12.5 to 13 nanometer in diameter. That will give the perfect specific absorption rate in the hyperthermia spec. Will actually reduce the size of the particle, which with the coating and with the peptide will actually have a very reduced size, which will actually prevent all a series of organ accumulation, which we have published in the previous work in terms of biodistribution, accumulation in the spleen and the liver. But because we do intratumoral injection, we're actually avoiding most of the problem of off-target accumulation.
Christoph.
Press it again.
Yeah. Sorry. Excellent talk.
Yeah, it's on.
It's on. So excellent talk. Two questions. First of all, is this the particles you will produce? Is this a medical device or a pharmaceutical agent?
Beautiful question. I was actually having dinner with Marisa last night. She said, "Come talk to us," because it's a matter of the mode of action according to the USA FDA. But according to the EMA, it depends on where the therapeutic window of your peptide kicks in compared to your mechanical accumulation of the particle intratumorally. So we have data to prove that the NoCan activate and gets activated later than the actual effect of the magnetic hyperthermia. So we believe it's a medical device. But it's all how you present, as you know, it's all how you present the case to the regulatory agencies and the convincing data that you support.
You know, we wish you all luck for your research, because the thing is, we are working also in a similar field. The thing is, if you do have the option to bring it as a medical device, it's much easier in compared to pharmaceutical agent. Maybe you have the chance, due to the fact that you are injecting the particles directly in the cancer, maybe you can overcome that this is not a pharmaceutical agent. But it's not a matter of the drug, it's a matter of the application form, I think.
Yeah, no, thank you. It's a very good point. As I said, we started from day one the discussion with the Spanish regulatory authorities because the application for clinical trial will be taking place in Spain because biopraxis is based in Spain, in the Basque Country. So that's where the effective dialogue has started from day one.
So I see that there are several more questions, and actually I also have questions for Adriele, but I'm afraid that we have to move on. We need to give enough time for the other three speakers. So thank you once again.
Thank you very much.
Now our next speaker